Hello, myself Dr. Avinash, and here we are going to discuss about smart study of biochemistry, ELISA part 1. So here we are going to discuss about what is ELISA. So in which we will discuss indirect ELISA. So first of all we will see what is the definition of ELISA. ELISA, it is an enzyme linked immunosorbent assay, and it is a common laboratory technique which is used for the measurement of concentration of analyte, usually antigens and antibodies, and now we are using hormones also in a sample or a solution. So here we prefer serum sample. Okay. Now this is the immunoassay, the method which is used for the detection of antibodies, proteins, peptides, and various biomolecules. Okay. Now, what is this enzyme linked immunosorbent assay? What is the meaning of this ELISA? So here, what is the meaning of sorbent? The sorbent, here it is a plastic surface, and on that plastic surface we attach antigen or antibody. Suppose you want to measure antibody on the plastic surface, we attach antigen, and suppose you want to measure antigen on the plastic surface, we attach antibody. So it is a well on which the plastic surface which is attached on it, and here it is called as a sorbent. The surface is called as a sorbent. Now the antigen here which is attached on the surface, which is recognized by the specific antibody. So it is a immuno term is used here, that is for the antibodies. So this antibody is recognized by the second antibody, the antibody which is recognized by the another antibody called as a second antibody, and which is a conjugated or which is attached with the enzyme. That's why it is called as a enzyme linked. And here we are using some substrate which is a colorless compound, and that substrate reacts with the enzyme to produce some colorful compound. So substrate, it is initially colorless, but by the action of enzyme on the substrate, it produces some product. And that technique which is used for the estimation of enzyme, hormone, proteins, everything you can, whatever you want which can be assayed by this technique, we can perform by using the ELISA kit. So this is a full form of ELISA. Enzyme linked means here it is linked with some antibody, immuno means antibody, and sorbent means it is a surface on which it is attached. Okay. Now what are the types of ELISA? Here we have 4 types: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So for our convenience purpose, first of all we will start with the indirect ELISA. So in indirect ELISA, you can see here this is a kit. Suppose you want to perform estimation of any hormones, there is a kit for the every hormones, that the specific kit, specific antigens, antibody, the combination it varies according to type of estimation. So here you have a well, there are 96 wells, and here we have the different bottles, the bottle for the enzyme conjugate, the bottle for the washer, buffers. Here we are using some standards, everything which is provided in the ELISA kit. So let's start with the ELISA. So this ELISA plate contains 96 wells. These are the 96 wells, and now we will zoom that. So after zooming it will be like this. Now here it is a clear, there is nothing in this well. Now on this well, suppose we want to attach antigen, we can attach antigen. On this well we can attach antigen, but as because of recent advances, the manufacturer provides antigen supplied from their side. So there is no need of addition of antigen. But suppose you want to attach antigen, so on well you have to attach antigen, you have to incubate for overnight, and on next day after washing you can use this plate. But because of the advances, the manufacturer provide antigen coated plate. Okay. Now here the specific captured antigen is immobilized on the high protein binding plates by overnight incubation. Suppose you want to measure HIV antibodies. So the antigen for this HIV antigen, these antibodies are immobilized on the high protein binding plate by overnight incubation. Okay. Then here sometimes there may be spaces. Here you can see the space, and on this space it may be occupied by the another antigen or antibodies. To avoid error in our estimation, it is blocked with the irrelevant protein, that is a albumin or bovine serum albumin. We can block it here. You can see this. Now for the convenience purpose I am not showing this albumin here. Now here we are adding sample. So here the sample is added. Okay, the sample is added here. Then here you have to incubate it for 60 minutes. Sometimes the incubation it may be for 30 minutes, 60 minutes, 90 minutes, depend upon the type of kit you are using. Okay. Now here we have already it is attached with antigen. Here we are. Now the sample contains antibodies, the antibodies that will bind to the antigen during the incubation period. Okay. Now there may be some other interfering factors are there. It is always present in the serum sample. So you have to wash it. We don't want that unwanted material for the estimation, for our assay. So you have to wash it. So here you can see this is a primary antibody. Now we are going to wash with a washer which is provided in a kit, or you have to prepare that washer. After washing, you can see it will be like this. Okay, everything is cleared. We have only antigen and antibody complex. Now here we have the other thing. We have a other antibody, that is a conjugated secondary antibody. It means it is an enzyme linked to the specific antibody, and here we have a substrate contains chromogen. Okay, you can see here this is an antibody which is linked with the enzyme, and that enzyme it is a horseradish peroxidase, or it may be a alkaline phosphatase, and here we are using substrate that is a tetramethyl benzidine. Okay. Now the antigen and antibody it is attached on the well. Now here we are going to add enzyme conjugate. The enzyme conjugate we are adding here. What is enzyme conjugate? It is the antibody which is linked with the enzyme that is a secondary antibody. So here we have added, and here you can see that this enzyme conjugate, that is secondary antibody, you can see here. So they will bind with the primary antibody, and here you can see the excess of secondary antibodies which is not attached to any antibody. So we can remove with the help of washer. So here there is additional step, because here we are adding secondary antibody, so we have to wash it again. So wash with a washer, and after washing you can see it will be like this. Okay. Now after washing we have antigen, primary antibody and secondary antibody, that is enzyme conjugate. Then now let's zoom this. Now after zooming in, which we have added substrate. Our substrate is tetramethylbenzidine. Now we have the enzyme. We have an enzyme that is a Horst peroxidase, and here our substrate is a colorless compound, that is a tetramethylbenzidine. Now by the action of enzyme Horst-Radish peroxidase, this colorless compound get converted into the colorful compound. So you can see it is a colorful compound by the action of enzyme Horst-Radish peroxidase. So here you can see, now if you have a more number of antibody, there will be more addition of the secondary antibody. So here there will be more color. So here there is a formation of color by the action of enzyme. Now because of the increased concentration of the antibody, if you have more antibodies, there will be more enzyme will be act on the tetramethylbenzidine, and finally we will get a more color. And here you can see again the revision is here. After in revision we will see we have antigen, we have a primary antibody which is attached, that is a serum sample containing antibody. Then we have added secondary antibody which is linked with the enzyme, and here it is linked with our enzyme with the help of biotin. You can see the rod like structure that your thin line which is there, and that thing which is that connects your enzyme and antibody. Then this is your colorless substrate. By the action of enzyme it get converted into colorful substrate, and because of that there is a color change. So here this is a indirect ELISA. Here you can see before addition of your serum sample, and this is after addition of serum sample. So here you can perform for the qualitative estimation, also for a quantitative estimation we can use this indirect ELISA